without further ado, I'd like to then introduce Jana Bromberg, who will be introducing Birte Hoecker. Jana got her undergraduate degrees in biological engineering, in computer science, basically, and biology from Stony Brook University. And then she did her doctorate and postdoc at Columbia University. She has a large program in Germany, and she has a large program at Rutgers, and she is one of the theme leaders of Theme 2. In fact, she is the leader of Theme 2. And so, Jana, it's yours.
Thank you, Paul. I'm extremely excited about today's lineup. I was also excited about yesterday, but today particularly. Particularly in the case of Birte, she works on things that are very near and dear to my heart. So it's my great pleasure to introduce Birte Hoecker. She's a professor of biochemistry at Bayreuth University in Germany. She earned her PhD in biochemistry from the University of Cologne in 2003. And then she worked on our shores as a postdoc at Duke in North Carolina. And she worked on computational protein design. In 2006, she started her own research group at the Max Planck Institute for Developmental Biology at Tübingen, investigating protein evolution and design. Her lab relocated to Bayreuth in 2016. Her main research interests are in the evolution and design of protein folds and functions, and her work has been recognized by a number of grants and prizes. The most recent award is for her and her colleagues an ERC Synergy grant to design autonomous motor proteins from non-motor parts, thereby exploring how complex protein functions can develop from simpler building blocks. And that's the part that I love. So Birte, we're really curious to hear what you have to say. Thank you for coming.
Thank you, Jana, for this very nice introduction, and also Paul and Vic for the invitation to speak here. And yeah, I'm delighted to tell you a little bit about our work on protein evolution, and this is always in together with protein design, because what we really want to do is understand how we can build proteins from scratch, and we can learn, I think, very much from evolution. I'm actually not sure whether you can see my pointer. Is that, maybe it's not working, but then I will just describe what I show you. All right.
I can see it.
Okay, good. There we have some options. Now, let's go ahead and start with what is really fascinating me and what's driving these kind of questions that we are asking. And that is that when I look at modern proteins, or actually modern organisms, they are really built from many, many different diverse proteins, and they are really diverse in protein folds and functions and structures. As you can see here in just a little cross section that was drawn by David Goodsell of an E. coli cell. And you kind of see here now a part of the membranes. You even see here the flagellum part. You actually have many, many proteins here in the interior that are enzymes for metabolic pathways. There are polypeptide chains coming here off those ribosomes, many proteins interacting with DNA, RNA. And so really what this is just showing is that we have very many different forms and functions. And basically the modern world modern life is really driven by protein functions. And so that's what fascinates me. And there are a couple of questions that we would like to address and answer. I'm not going to be able to fully answer them in this talk, but I want to yeah, I think they're basically overlapping quite a bit with some of the things that you're looking at in the ENIGMA projects. So on the one hand, the question is really, how did all of this diversity emerge and then continue to evolve? What can we learn about that? Is actually something that has nature really sampled all the possibilities of proteins? And can we learn something from this to design sequences that will also fold into functional proteins, or proteins that are just as functional as the natural ones? That is sort of the ultimate goal. So let's look at really what makes up proteins and how what the difficulties are. And that is that really only a random fraction of proteins or polypeptides will really fold into a functional form. So illustrating this here by this more polypeptide chain that's more supposed to be random, that will come together, form a compact structure. And for most functions, you really need this compact structure. So in order to, for example, have catalytic activity, you have to have a folded state. And so you have to form binding pockets, active sites have to come together in order to be able for this protein to catalyze, for example, certain reactions. So really, this is a problem that proteins have to find this structure. And so not every peptide will do this. So this is a conundrum then. How is nature dealing with this? Also, there's an additional problem. Folding not only is complicated, but it's also easily disrupted. So there are also many diseases associated with misfolding. And so this is what nature has to deal with. And so how does nature circumvent this whole folding problem? Well, what it does is it reuses. So once it's found one solution, it actually takes this solution and uses it again. So basically, nature bypasses the folding problem by taking a piece that worked and then using them in different contexts. And so, for example, here you have this, you have the main blue domain, and you can see this in natural proteins now, that there are, again, always these similar domains, and then you have different accessory domains that are important for particular functions. And so the units that are very obvious to us are domains, and domains are the unit typically described as the units of folding. But if you think about it, even those domains are already too complex to have arisen de novo. Domains are often 100 to 200 amino acids in length. And so if you think about all the possibilities that you have, if you have 100 amino acids just randomly associated, you wouldn't necessarily, well, it would be really hard to get a folded protein. And so even on the domain level, this is not, they must have arisen in a different way. And so while we on the one hand see that duplication and recombination and reuse of domains is a driving force in evolution, we actually, when we look closer, we can also find subdomain fragments that have been recruited over and over again in order to form the domains that we observe today. And so here I have just my favorite Tim barrel domain here, still as a blob. I will show you the structure in more detail next slide. Associated with two accessory domains, for example, the pyruvate kinase or enolase, you have another domain coming up there. The Tim barrel is supposed to be a very compact structure, but also in there, and I will show this to you in the next slide, we actually see that they can be built up from smaller units. In fact, Tim stands for, or the structure is a beta alpha 8 repeat. So you already hear that there's secondary structure elements that are repeated, and there we actually also see that there's smaller units that could have been used to build up the Tim barrel. And what's much more obvious is in these repeat proteins like TPR repeats, armadillo repeats, ankyrin repeats, and many of these you actually see it's much more apparent these kind of units that are used again and again. And that's something that has been explored on the Tim barrel, or here shown as a beta alpha 8 barrel. So you see here in the center, the 8 beta strands that form the inner core of the barrel, and it's not empty. There's actually side chains interacting here at the center. And that this is surrounded by 8 alpha helices. And that is a very prominent fold in nature. Many enzymes have actually this fold, and you find it on almost 10% of all known crystal structures you find in Tim barrel. And here, is actually 1/2, at least in 2 proteins. It's very obvious from histone biosynthesis pathway where you see this very obvious twofold symmetry. Then, for example, in this beta trefoil structure, there are 3 units of this blue part here that are repeated. These are propellers, propeller blades that can come in many different blades that can come together to form different types of propellers, or for example, beta hairpins, such as here in these membrane beta barrels. And so these are units that have been used by us and by others in order to build proteins again by duplication. So for example, what I've done myself is actually taking 1/2 of this Tim barrel and just taking the same one and duplicating on the genetic level, and then expressing this protein and looking at the structures. and analyzing the proteins. And so we could duplicate one half, this is the C-terminal half of the his-F protein in this case, which we duplicated, and then we could get a well-folded protein structure. There's a couple of things that we have to adapt, so but then we could solve also a crystal structure and see this symmetry, and we could show that you can take one half, duplicate it, optimize it, and actually again yield a full TIM barrel. Similar things have been done with a beta trefoil by Blaber, and they have built also by triplicating. Good. So duplication is clearly a way we can build up structures, but we can also recombine pieces that are maybe very similar. So for example, for the TIM barrel, we could take one half from this his-F protein, and we took the other half from a related protein from histidine biosynthesis pathway called his-A, and we recombined those two halves, and actually we got a nice new TIM barrel, which folded nicely and behaved well, and we could evolve a functionality on top of this, just starting from the modern fragments that we recombined. So recombination is another mechanism that you can use in order to build up this complexity. But so far, this has all been done just in single folds, right? I take one piece and I duplicate it, or I recombine it, but within the same fold. So what about different protein folds? What if I compare two different protein folded proteins? Such as, for example, this his-F TIM barrel, as I've shown you before, and now a flavadoxin-like protein, the chemotaxis response regulator QI, for example. This one is also a beta-alpha fold. It has 5 beta strands here in the center, and then it's sandwiched between helices here on the left and right. These parts actually, these proteins actually have some parts that look very, very similar. For example, here, this part in green, these 4 beta strands and 3 helices, they look very similar to this gray part here in the his-F protein. And so we asked ourselves, couldn't we maybe just swap these parts? Can we take a piece from QI and place it into the context of the his-F barrel? And that's what we did. Genetically, again, we cloned the sequence, we expressed the protein in E. coli, and we could actually solve the crystal structure of this protein that E. coli produced for us. And we call it QI his-F. It's our fold chimera, so we call it. And we got a very nice barrel structure, but it's not the typical beta alpha 8 as we expected. It actually is slightly different. It, in fact, has here at the core a 9th beta strand. So let me walk you through. We have the N-terminus here. We start with the first beta strand from QI in green and continue the polypeptide chain. And then here in blue starts the part from his-F. And then we go all the way here to the C-terminal end of this part. And we have something that I colored here in red. And that's the 9th strand. It originates from residues in his-F that are at the C-terminal end, but that don't form a secondary structure element in his-F. And actually also some residues that we had added from the his-tech for purification purposes. So these residues are not typical for a beta strand, but they kind of snuck in there and placed themselves in the core of this protein and formed a 9-stranded barrel. You can see it maybe more easily from the side here, where this 9th beta strand is sneaking in between the first and the second beta strands of the QI fragment that we used. What's also interesting here is that we have a binding site that we carried over with the his-F fragment. So his-F has binds a biphosphorylated substrate, and one of those phosphate binding sites is still present. And in our crystal structure, we actually have a sulfate bound at exactly this position. So we are also carrying over that functional site. Right, this was actually the work of a summer student, Tanmay Barat. And so just to summarize, we've taken this piece from QI. We actually have done it also from the nitrite response regulator NAL, and we could make chimeras from this. From NAL, his-F, we don't have a crystal structure, so we don't know how exactly it looks like, but it was also a well-behaved protein. So the next question we asked was, what happens, why do we actually have this 9th strand? We didn't expect that at all. It really makes a difference in terms of the integrity of the core. And so we first of all asked, how far away are we from making a typical beta alpha 8 barrel? Because if we cut away those residues, that form this red beta strand, then the protein tends to aggregate. And so we thought something is not fitting quite right at the interface of those two fragments. And this is not surprising. These are, if they're even evolutionary related, which at that time we didn't know, it must have been a very long time. So just taking from modern proteins two fragments, you wouldn't really necessarily expect that they fit perfectly. And so we use computational design to actually optimize the interface when this beta strand is left out. And we used Rosetta design and introduced 5 mutations at the interface between the two fragments, 2 on this side and 3 on this side. And these 5 mutations, they really made a very nice new protein where we could solve also the structure of this protein. Interestingly, we couldn't get any crystals, but we could solve it by NMR because it was very, very stable at high temperatures, and so it was possible to solve the structure. And so this work was mainly work with Simone Eisenbeiß, where we collaborated with Jens Meiler for the Rosetta design. All right, so now this protein is well behaved, but what can it do? So we actually, as I told you, we carried over this phosphate binding site from his F. So we thought maybe we can do something with this. And we looked at a ligand that is similar, not the same, because his F would require 2 phosphate binding sites, but we took a related compound from tryptophan biosynthesis, which has 1 phosphate binding site, and then we can nicely measure binding by a fluorescent signal. And we could actually then, we had already some binding. This is the red curve here, the binding curve. But we could improve it with 2 single mutations that we placed rationally based on knowledge that we had for binding of this compound of the natural protein, which is shown in blue, the binding curve. And so 2 more mutations bring binding of this compound in this chimera to a similar level. And so we would argue that with this evolutionary path or design path, recombination, a couple of mutations at the interface, and adaptation of the binding pocket, you get to something that could be a competitive protein in the natural world. And so we think that in some ways our designs are mimicking possible evolutionary events, that when you take fragments, and even fragments from modern proteins, so we didn't take anything that's ancestral, but modern proteins, and then you combine them, you get something that actually we call a hopeful monster. This is a term from organismal evolution, but this is actually a new protein fold that has not been observed, as far as we know, in nature. No 9-stranded barriers are known. So this could be even starting points to new folds. But then in many cases, you probably, once things are not fitting quite right, a few mutations will lead in a futile cycle back to something that looks very much like a starting protein fold, and can even be, and might actually even have a functionality like binding and so on. So now when you look at this, the question is, how can I differentiate something where this has happened, where maybe a flavadoxin part has recombined with a TIM barrel? How can I now identify this afterwards? You know, all TIM barrels look like this, so maybe this has actually happened. Can we find out whether these 2 folds are truly evolutionary related? Because when we started off, we didn't know that. We just saw the structural similarity. And so if you want to say something about the evolutionary relatedness, you really have to find sequence evidence, because structure, if the structure is the same, this could also be just convergence. And so what we tried to do is then compare proteins from different folds. So just to bring you all to the same level here, probably you know, but when we have all those protein structures, we put them in, try to organize them, so, for example, in the Scop database or CAS database. So we are using Scop very much. And so there you have the level of the different folds, like the beta alpha 8 barrel, as they are called in Scop, and the flavadoxin. And underneath you have different superfamilies. And both of these folds are actually superfolds. They both have many, many different superfamilies. The TIM barrel has 33, and the flavadoxin likes, they have 15 different superfamilies. I just show 2 as an example. And so usually when you look at a superfamily, you have similar structure, similar sequence, similar function, and this is evidence for homology. But what we actually want to look at is now at this level between different folds or between different superfamilies. All right, so Simple PSI-BLAST will not really detect this, but there are very nice tools out there that were developed by Johannes Söding that use hidden Markov model profiles comparisons. And so we made these HMM profiles of all of those 33 superfamilies of the tin barrels and the 15 of the Flavadoxin-like fold, and compared all against all. And I'm trying to now put all of that information in one slide, so bear with me. So we have now all the 33 different superfamilies here with the numbers. The order doesn't really, you don't need to really look at. It's all the 33 are there. 15 superfamilies from the Flavadoxin-like fold. And now I'm going to draw a line whenever we have a bidirectional hit with this HH search program with a certain p-value. And so
I'm sorry. Hold on, Birte, you should explain very, very, very briefly what a hidden Markov is, because I don't think people understand this. Just this is okay. Go on.
So when you do sequence comparisons, you can compare one sequence against another sequence directly, or you can make multiple sequence alignments, and then you will get a lot more information within this multiple sequence alignment that you can compare against another sequence, or you can compare these these multiple sequence against multiple sequence. So in this way, you can make all of your searches much more sensitive, and that's what you basically do in these searches. It's profiles against profiles, what you basically do in these HMM search method from Johannes Söding. I hope that abstracts it.
Yeah, thank you. I mean, we start with no observation at the beginning, so you don't have a zero. So you're just looking at what are the most relatable sequences through.
Right.
That's what the hidden Markov is. Really, most relatable.
Mm-hmm. But you basically, what you can do is now by doing these profiles against profiles is that you can also tease out things that are much further away. So basically, if you just take a single sequence, you might not actually see these things. So this is a way to find remote homologs.
Thank you.
Mm-hmm. Yeah. So if we just start out with the Flavadoxins here on the right-hand side, I actually only can draw a very few lines. So there's only a couple of them that really show any similarity at the sequence level. And that means that the Flavadoxin-like fold, we don't know. Maybe it had actually evolved multiple times. If you look at the tin barrel, the picture is totally different. You see many, many connections. And a lot of these were known before. So there was already 10 years before, 2004, there was work by Janet Thornton and her group, and also from Per Borg's group. They had already related half of those tin barrels. And now with these profile comparisons, you can actually connect almost all of them. And there's only 3 where we don't draw lines at this level, but it's also, we don't have many known structures of these superfamilies. So maybe the data is not there yet, but this looks actually as if the beta alpha 8 barrel superfamilies, that they have a monophyletic origin and that they might have only arisen once. And that's in great or stark comparison or contrast to the Flavadoxin-like fold. But what's now more interesting actually is when you look in between, whether we get hits in between those. And there we have many hits between 2 of those Flavadoxin superfamilies and about 10 of those tin barrel superfamilies. So which are they? Here on the tin barrel side, they are all the phosphate binding barrels. So there's a whole big number of superfamilies that have substrate that have a phosphorylation site. And so they have this functional similarity there. And then here, those 2 superfamilies from the Flavadoxins, it's on the one hand, number 1 is where our QY came from. So these are all the response regulators. It's also a very large superfamily, and which we actually used in building our chimeras. And then we have the vitamin B12 binding Flavadoxin proteins that actually even have more hits or hits to even more tin barrel superfamilies. So we have very many connections here, which was very interesting to see, especially when you see that the Flavadoxins themselves don't even share that high similarities. So that was overwhelming. So we thought, let's go one step further and not only look at similarities between proteins where we know the structure, but go to sequences where we don't know the structure and see whether there's something that's intermediate, something that's in between, and where it would be interesting to solve the structure. And we found a whole family of sequences that are in between those two superfamilies. And we picked, so it was a whole group in different organisms. It's not just an isolated case, but it was many of them. And so we solved the crystal structure of one of them. And they're always associated as an N-terminal domain to a different domain. And so this one is called NTM0182, and it's from Thermotoka maritima. We don't really know its function, but we solved the structure of this domain. And in the crystal, it actually forms a swapped dimer. So swapping is not unusual. You can have the swapping happening. So here's one beta strand that is coming off from the other domain. But whether this is natural, we don't know, because when we isolate the protein and purify it, we actually observe monomers and dimers, but it's only the dimer form that crystallized. And so basically this would be the topology on the right hand side. And what you see is that you have this typical beta alpha modules. It looks very much like the flavadoxin here at this part, but it has a 6 beta strand, which flavadoxins don't really have. So once we compare all of this, we actually see here on the left-hand side the closest TIM barrel, on the right-hand side the closest flavadoxin protein, in the middle the structure of our TM0182. And when we focus the sequences where we have the highest similarity, basically it's the highest at not the full length, but mostly at this fragment, which is about 40 amino acids in length. And it's an alpha beta fragment. And interestingly, this is in the area where in the TIM barrel we have ribulose phosphate bound, and in the flavadoxin-like protein we have the adenosyl arm of the B12 bound. The ligands have, I mean, they both have a ribose no, they have a sugar moiety and they have similarities, but they are still quite different in the way they bind. But it's interesting that this fragment is exactly where the function is. So maybe there is some context why this is conserved. And we don't really know what this protein is binding. It wasn't binding ribulose phosphate or B12, but there might be something else binding there. Okay, so the other thing that you might see is that when you look at the beta sheet here in the flavadoxin, it's a fairly, it's not as curved as when you look at this as our sort of intermediate and our TIM barrel. And so that's the other thing that we observe, that we have a little bit more curvature here, and we have the 6th strand. All right, and now the other interesting thing was that we found even a smaller piece. Remember that in the beginning I was telling you about half barrels, but this is actually an indication that it might even have been a quarter barrel that could have been the ancestral unit. And you actually find in the TIM barrel 4 times this fragment. You can overlay this quite nicely. And that's interesting because we actually also have, and this is now sort of an excursion, just a short one, to some Denovo design that we have done in recent years with David Baker and Alejandro Fernandez Velasco, who is at UNAM in Mexico. And we actually have designed a TIM barrel from scratch, which has been a long design goal. And here we actually use this 4-fold symmetry. Just saying we wanted to make the design goal simpler, and I'm just going to go through this fairly quickly. But here you have the barrel shown, and you see how the beta strands are tilted. And so we started with the beta strands. We looked at, we said, okay, the beta strands have to be looking this way. You always have one residue looking outwards, one inwards, outwards, inwards. So you have the residues in the beta strand always looking in the opposite directions. And so you have a pattern for these. And then you flatten it out. So you have your first beta strand, second, third, and so on. And then this is the first one repeated. Okay, so therefore we knew what the geometry of the central beta barrel should look like. But how do we connect these? And how can we idealize this in order to make it 4-fold? We actually defined always 2 beta strands that are parallel to each other and connected them with helices, and you have to connect them a little bit longer for the first 2 because they are even barrel facing pleats. So they actually, where you always have this as a starting point, a residue facing inwards. And this is something that we took from the rules that were defined by Kruger et al. On how you connect an alpha helix with a beta strand. All right, and so then you know how this how the length of the alpha helices should be, because they need to be long enough in order to connect those beta strands. And this one is then, the second one is tilted and shorter, and then you basically just repeat. And so we use the Rosetta design software in order to find conformational free energy minima and find the best sequence for this. And so it was a fourfold repetitive sequence. And to cut a longer story short, we actually were able to solve the structure of one of those designs. And we analyzed the protein. It's unfolding with guanidinium hydrochloride, looks very nice. It's not, so we have fluorescent, and so when we look at secondary structure loss and loss of tertiary structure, it's not right on top of each other, but otherwise it's both very nice indication of a well-folded protein. We have thermal denaturation shown here, so it's highly stable, so up to 80 degrees before it really fully unfolds. And when you refold, you get again a CD spectrum that it looks exactly the same as before the unfolding. So it's a reversible process. So this is a very nicely behaving protein, and we could solve the crystal structure, with is shown now the backbone in blue on top of the design in pink. And at least in the cartoon, you see that they look nicely on top of each other. And when we now look at the ribbon here of just one alpha beta module, you can see that this is also very nicely on top of each other. So this was a Denovo design of a fourfold symmetric TIM barrel, showing that you can take this fourfold symmetry in order to build a new TIM barrel. You might now ask, is this really new? Because I told you before that all of those TIM barrels, they are related, seem to be related, they have some sequence similarities. So how does this Denovo TIM barrel compare? And so when we do sequence comparisons, we don't find any similarity. We don't find anything when we compare, when we do position-specific iterative blasts 3 times, which is usually where you would start to see hits. We don't see any hits to any TIM barrel. Only when we do these hidden Markov model profile comparisons, we start to see some similarities. And then you can take these similarities and map them in a cluster map. So basically that's what I'm showing you here. Every dot is one TIM barrel, and we color code them based on which superfamily they belong to. And so you see that many of them cluster when they come from the same superfamily. And there are connections basically whenever they are shown in the 3-dimensional map. But basically what a cluster map does is it's a different way of showing phylogenies of large numbers of sequences. And basically what you see is that all of these TIM barrels, they have some connectivities, most of them. There's one superfamily that's out here, one sequence that's out here. But where is our Denovo TIM barrel? It's up here. And it really only shows very few connectivities. So what we think is that with this new Denovo TIM barrel, we are actually finding new space, new sequence space, and exploring new sequence space. And that would at least answer one of these questions at the beginning. I don't really think that nature has sampled everything. And there are still areas of that space out there that we can sample and maybe actually access with Denovo design. And we have continued now on this and built, as Sergio Romero has, he has built a whole family of these Denovo TIM barrels, and we're exploring now in that direction as well. So to have a short summary in between, with these sequence comparisons, we actually find evidence for homology between folds, and we can identify subdomain-sized fragments, such as these alpha beta fragments in the TIM barrel Flavadoxin protein. With the Denovo design, I've shown you that we also can build them de novo, and then we can also recombine fragments. And I think this is really giving us a feeling that we can use these pieces as building blocks and in order to build more complexity. And that was the idea that we had for our last ERC, which we actually called Protein Lego, because we want to use these fragments in order to build up complexity in a Lego-like manner. And we thought that this really invites a general approach of using these fragments to build new proteins and, if possible, enzymes, but that's a different story. So how would you go about this? How can we identify useful parts for the design by recombination? Well, we can do it just as I've told you before, by these sequence comparisons, as I talked about the TIM barrel versus Flavadoxin and the fragments that we got out of this. And now we said, let's do this in a global fashion. Let's do this now of all the proteins that we know and that are in our databases. And so we started building a fragment database We call it the fold puzzle database, short fuzzle. And what we put in here is, on the one hand, these profiles that we generate when we compare all against all. So, for example, you have one sequence against another one, and the yellow part here, this is the match, and that's the hit that we store in our database. And then we actually connect this to the structural information, where we also do superimpositions and take a look whether these actually also structurally superimpose, and then we also deposit this in the database. And there's many, many hits that we get. And so that means there's lots and lots of pieces that we have there that we could theoretically play with in order to build new proteins. Let's just briefly look at what is in this data set. This is just a different means to show all of those connections in a network. So this is a network representation where every dot or each node is a domain that is colored based on the Scop class, according to the Scop class as shown down here. And you can see already all alpha/beta proteins, they start to cluster here in the center. We have many, many all alpha proteins that are clustering here. And then the other ones are more smaller islands. So we have one major component that really pops up here, and that's zoomed in on this side, showing also which superfamilies or which folds they connect to. So the C1 is the Tim barrel that I showed you before, and C23 are the Flavadoxin that we talked about. And so they are, for example, here you have Rossmann folds, many of them. The Rossmanns or Rossmannoids are in here, and then there's many of the all alpha proteins. Just to show a couple of examples from this analysis. Here, the first and the second cluster, they are examples of the Tim barrel and the Flavadoxin, as I've shown you before. And interestingly, there are areas where we have sequence similarity, but they are not in the structural similarity visible. For example, this helix here that's shown in red, the sequence is very similar to the one here of the Tim barrel, but then when you superimpose them, you see they're in different places. And then interestingly, the same proteins pop up again, but it's the second half. So here we have the one half of the Tim barrel, here we have the other half of the Tim barrel, and you have a separate hit. Also, for example, here shown is a Rossmann fold. The C.2 is Rossmann folds, very, very large class of or group of proteins as well, together with the formyl transferase-like fold here, the C65, where we have a common fragment. And then here's just an example on the bottom from the all alpha. So this is the most known fragment, most frequent all alpha domains, which are DNA, RNA binding, 3 helical bundles with a lambda repressor that are connection, where we basically have a smaller fragment here. So you already get a notion that the fragments can be of very different sizes. So we start from 20 amino acids, but they can be up to 120. So they can range a large area, in fact. If you want to search through them, this is now possible through our full puzzle database, where you can actually, to the web interface, if you go to this website, then you can browse this yourself and just look for the proteins that you are interested in, whether you find actually interesting fragments there, and where you can visualize this on the structural level and on the sequence level, as well as with the clustering that I showed you before. But let's go briefly to maybe a simpler way to visualize this. This was a way I've done it before. We had this web interface, and that was just listing here all the Scop superfamilies of the C class, which are the alpha/beta against each other. And because I told you about the Tim barrel and the Flavadoxin, all of the sequence comparison that we did is contained in this data in this little red box. And if you look at all of those other red, blue hits with very high probability hits, you see that there are many, many pieces. And so these are these many things that you can use in order to build new chimeras. And that's something that we've started to do, for example, for all the Flavadoxin likes, which are in this line. For example, you find the paraplasmic binding proteins, which bind sugars, amino acids, and so forth. And here you have a very clear similarity also on the structural level, where you see similarity of this is actually now also QI, and which we placed into a leucine binding protein and could make a chimera with a paraplasmic binding protein. And for example, the heme D-like. Heme D-like are the uroporphyrin synthases. They bind a precursor of heme, and there it looks as if this whole bottom lobe is the flavadoxin domain. And so that's something that PhD student Saknikte has done. She actually placed from methylmalonyl-CoA mutase this almost a full domain, not entirely, but almost, into this uroporphyrin synthase from human, and recombined it and could solve the crystal structure of this new protein. And we have some indication that the vitamin B12 binding of this methylmalonyl-CoA mutase is also transferred to this new hybrid protein. So we can build chimeras, but what does it really tell us about the evolution of these folds? Interestingly, this data in the fossil database can also give us a lot of hints of how maybe the evolution has happened. And in this HEMD fold, it's interesting because what we see here before, that we have a structural similarity on this lobe, is not reflected in the sequence. In the sequence, you actually have similarity across the two lobes. It looks like a duplication of a flavadoxin, but one that has a swap. So if you had sort of an opening of this, this one part through insertion of a short segment, that maybe this can relocate and then opening up hydrophobic space so that it might actually dimerize. And then once you have a duplication, you would lead to a full HEMD-like protein. Let me show you this in a little more detail. Look, we have here now the flavadoxin at the side, and we have the HEMD here. And the idea is that by insertion here at this loop, this would open up and then could duplicate. And if you then have a gene duplication event, this would lead to HEMD. We actually tried to do this in the opposite way, to reconstruct this, this evolutionary event, starting from the HEMD. And here we split it in two halves, and we made a HEMD C-terminal half, which was kind of unstable. It was starting to aggregate. But then when we extracted these 5 residues from this extended loop, we actually suddenly had a very nicely folded protein, and we could solve the structure by NMR and ended up with a typical flavadoxin protein. So you can see that you easily can take a modern HEMD-like protein and reconstruct it by just taking a half, taking out these 5 residues, and you end up with a very nicely folded flavadoxin protein. All right, so we've explored a couple of things, starting mainly from the flavadoxin protein towards the TIM barrel, towards the PBP-like, towards the HEMD-like, and we're working with Rossmann as well. Well, what have we? Well, we have learned quite a bit from this, but what is still missing? At the moment, we're looking at structural level, and I think what's really important is to also include ligand information. And we have now at least done this and placed it into Fuzzle, and there's already a new version online. Paper should come out soon. It's called Fuzzle 2.0, and there you actually now have also the information whether a ligand and the structure is associated with a fragment that we think is an original homologous fragment. And the other thing is that we would like to automate this process of chimeragenesis, and that's something that Noelia has now done, a postdoc in the lab, and she has made it that you can now automatically fetch from the Fuzzle database these networks, and from the networks you can choose pairs where you want to build chimeras. For example, she's in this paper, she shows it for the Rossmann versus a P-loop, something that Denny Tawfik has worked with quite extensively. And then you can build all the possible chimeras. You can score the chimeras by their energy and analyze the structures based on hydrophobic networks, clustering, salt bridges, and so on and so forth. So if that's something that you're interested in, the code is available here on GitHub. And that's a tool that maybe is going to be useful for you as well. And these analysis that's at the lower end here for the structures, we actually also put this into a web server that's called the Protein Tools web server. So if you just want to analyze of one of your proteins, the hydrophobic clusters on hydrogen bonding networks and so on and so forth, then maybe this is of interest to you as well. It's under Protein Tools unibayreuth.de. All right, I think I'm running out of time. I'm sorry I talked too long, so let's wrap up. The take-home message basically is that the modern diverse proteins that we observe today, they, to me, it seems very clear that they evolved from smaller fragments, from subdomain-sized fragments via co-option, whether it's through duplication or recombination and then diversification. And we can identify these fragments by sequence comparisons, and you can now search them in the fuzzel database. We ourselves use these fragments to construct new proteins and think that this is a way you can really directly link protein evolution with design. And at the moment, this is what we're still working on. And as you already said, our next big goals will be going towards protein motors, but that's what the future will be. So I'd like to thank my team. The current team is shown here, but I've already had the pictures and names of the people involved in all of that work before. Also, I'd like to highlight collaborations with Steffen Schmidt on the fuzzel database, Christian Schweimelz with NMR here in BioBite, but Murray Coles was our NMR expert that we worked with at the MPI, David Baker and Alejandro for the design of the Tim Barrel. And then I'd like to just mention also our consortium that we have here so that we have funded to the Volkswagen Stiftung under the name of LIFE. What is LIFE? And so I think you've seen, or you had Andrei Lukas visit you just a few weeks ago, I think. So he's part of our consortium, as well as Nir Ben-Tal, Rachel Kolodny, and from Weizmann Institute, Sarah Fleischmann. And unfortunately, I mean, Danny Taufik was part of our team, and we will very much miss him because he's been, just a great person to talk to, a great scientist, and a very good friend. And it's been really a pity that we can't work together anymore on how proteins emerge and continue to evolve, but we will try to continue with him in mind. So thank you for your attention. I'm at the end right now.
Thank you, Birte. I think we are over time, but I will ask my postdoc to move down a little bit and everyone to have some patience with their break. So we have 3 minutes for questions. The first one was from Vic. Do you think which one came first, Flavadoxin or the Tim barrels? And was there an ancestral protein, or is that just in the intersection of the modern folds?
So I don't think we can say what came first. Both of the folds are considered to be very old. I think Tim barrels and Flavadoxin are supposed to be among the 5 oldest folds. So I wouldn't really want to say which way around. The one that we call intermediate, I would also say this is a modern protein, so I can't really say which one was first. I rather imagine that it's some of those fragments that were more ancient and then have led to all of these different proteins. Cool.
And then Drawer asks if the Denovo, regarding the Denovo Tim barrel, if it is a sample from a non-natural sequence space, what do you think will happen if you subject it to directed evolution? Will it provide more samples from the new space or fall back into the natural space?
Depends. I think you're quite far away, so it probably will be more in the vicinity. It depends on how far you allow it to mutate. But I think you can. I mean, that's going to be the next thing. We're working on extending this. At the moment, it's more like a rock, right? Because it's idealized as short loops. In order to really functionalize it, I think we need to build in longer loops, maybe more flexibility and whatnot. But I don't think we will really go away so far from that sequence space that we have there. So I would rather think we will stay in that vicinity. At least that's what you also see when you do directed evolution, that you kind of sample around it, but you don't really do very big jumps. And I think that's what the Denovo design can actually give you, bigger jumps into different areas. And then you can start exploring it with all these other techniques.
Interesting. I would like to discuss that further, but I will take one more question from Diego because it's in line with what I've been thinking about. Do the fragments of the fuzzel correlate with exons? I wouldn't expect them to, but do they? No, not obvious. No, not very obviously. They don't. No. But this is a typical thought that many people have, that maybe this has to do with it, that it's exons that evolved in this direction. This has been discussed, but we don't really see that. So the reason why it's of interest to me, and I'm going to ask my question, prioritize myself here, but the point is we have a way, we have designed a new way of comparing structures and then using the sequence similarity in the structural alignment in order to infer whether things are related or not. And I find that that is a lot more open in the sense that you can find a lot more hits in a lot more places than what you would expect by going the other way around, by first going through the profiles and then comparing the structural similarity. So from that perspective, first of all, have you thought about doing that? And second, have you considered the similarity, sequence similarity in structure space of your fossil domains?
So basically, Rachel Kolodny, who's a partner in that in our consortium there, she had started by just basically doing this on the structural level, and we had lots of discussions on this. I think it's hard to really then infer that things are really evolution related if you don't take the sequence into account. If you start off with if we start off with a structure and then find sequence similarities, then I think that's probably just fine as well. But shouldn't you find these things both ways? Shouldn't they overlap? I would expect this, because if you find the sequence similarity as soon as you superimpose these things, I think you should also find them on the sequence level. Because in the end, yeah.
Sorry, that isn't, I'm sorry, I maybe haven't been very clear. On the structure level, things are not consecutive. On the sequence level, you could find things that, yes, you could use profiles and you could find things that are consecutive amino acid similarities, but anything that's a flip is not going to come up no matter what you use. Markov models will not pick up on structural flips either, right? So there is going to be a lot of overlap, I suppose.
But can you find even an evolutionary scenario that is fairly simple to evolve from one of those to the other? Because that's the thing. For example, the KH domains are an example like this, where you find structurally very similar things, but it's very complicated, the scenario that would lead to such a different arrangement. So if you have too many fragments, that means that there have to be many different steps. And I think that makes it more unlikely to have happened in a certain way. And that's why I would be careful with this. I mean, I think those are very interesting cases, and maybe you even find cases then that are very unlikely. But if they have, if you have to swap too many things, that's many events that have to have taken place. And it can have happened, but I think the more steps you need, the more unlikely it will be.
Okay, I agree, but I'm sure that we can have a really long conversation on that topic, and hopefully you stay.
I'd be happy to do that later, but I'm very sorry that I over talked.
No, that's a wonderful presentation. Thank you very much.